My name is Cameron Clark. I'm a student services contractor in EPA's Computational Toxicology, National Center for Computational Toxicology. Today we're going to be hearing from Dr. Tamara. She's a biologist in EPA's National Health and Environmental Effects Research Laboratory, and she's going to be talking about investigating chemical microbiota interactions in zebrafish. So if you have any issues with seeing or hearing the presentation, please speak up on the line or type something in the chat box, and we will try and address that. But with that, I will go ahead and pass the torch here. All right, you should be good.
Okay, thanks. And I'll see the chat come up on.
Yeah, so if anybody speaks up in the chat box, we'll just see a little message come up in the corner there. I'll try and keep an eye on that, so don't worry about it.
Okay, well, first, thanks so much for the opportunity to present our research. As Cameron said, my lab's working on looking at how microbiota might modify the toxicity of environmental chemicals using a zebrafish model. I'll start with a brief background on microbiota and using zebrafish for this type of research, and then I'll give examples from 3 case studies we've been working on, looking at triclosan estradiol, BPA, and a suite of BP replacement chemicals. And I'll summarize the findings and then leave you with 2 big challenges that I see for how to move this research forward. So microbiota consists of all the bacteria, viruses, fungi, and archaea in the body. Colonization begins at birth and continues throughout life, so this is a highly dynamic system that is quite variable from person to person and even within an individual over the course of life. Colonization with microbes is increasingly understood to be required for a number of developmental programs to develop both organs and systems, and here the classic example is the immune system, which relies on colonization by non-pathogenic bacteria for normal development. And increasingly, we're beginning to appreciate this link between microbes in the gut and the brain via the microbiota gut-brain axis. So this connection is mediated bidirectionally, both directly via the vagal nerve, which innervates the intestinal tract, and indirectly via the secretion of soluble factors by microbes that colonize the host, that move into the host circulatory system, penetrate the blood-brain barrier, and exert direct effects on the developing brain. And we know, primarily from mammalian studies using gnotobiotic or microbe-free mice, that colonization status modifies a number of neurodevelopmental events. So it seems to be required for things like myelination, blood-brain barrier development and maintenance, neuronal migration, microglia activation, and that's just listing a few. And we know increasingly from epidemiological studies that imbalances in gut microbiota composition are associated with a number of behavioral disorders. And the classic examples here are mood, anxiety, and autism spectrum disorder. So again, these are associations, not causal linkages. And to date, microbiota has not been assessed as a modifying factor for the developmental neurotoxicity, or DNT, of environmental chemicals. So that's kind of where we started off. And there's 2 theories about how microbiota might modify chemical toxicity, and to introduce those concepts, I'm anchoring them to the adverse outcome pathway, the generic adverse outcome pathway framework that I'm showing here, which many of you on the phone are probably familiar with this, so I'm not going to belabor this slide, but essentially it's a framework to connect a xenobiotic interacting with a biological substrate to perturb biology at higher and higher levels of organization, ultimately leading to adverse outcomes that are of regulatory importance at the level of the individual or the population. And some of the adverse outcomes that the agency is interested in is listed on the right-hand side of the slide, including DNT. So the first theory about how microbiota might interact with chemicals is toxicokinetically. So here the idea is, because the microbiota system is so incredibly diverse and dynamic, the genetic component of this system is amazingly vast, and it's been called our second genome. Oh, okay. And so the idea here is And I'll just say I'm having a hard time reading those pop-ups, so if you have questions on the line, I'll answer them at the end. Okay, so for the toxicokinetic hypothesis, the idea here is that these microbes encode proteins that can biotransform chemicals to either bioactivate or detoxify them, and in that way change chemical toxicity. The second hypothesis relates to dynamics. So this is basically predicated on the idea that because microbial colonization is required for normal development of the host organism, if the organism experiences chemical insults during critical windows of development that prevent normal colonization or impair it in some way, you can have a secondary adverse effect on the host. So the hypotheses that are driving this research are that host-associated microbiota either modify the toxicity of environmental chemicals by performing biotransformations, or that microbes are a target of chemical exposures during sensitive windows of early development. So this relates to the toxicokinetic and toxicodynamic hypotheses. So we're using Zebrafish as a model system to get at these questions for a number of reasons, and the first two bullets really highlight the benefits of using this model for developmental toxicology research. Zebrafish develop external to the mother, so you can directly dose the developing embryo. You do not need to dose the dams or the moms. And development occurs rapidly. So this organism goes from a single fertilized cell to an organism with a body plan with a beating heart in just over a day. Organogenesis is complete by 3 days of life. They contain every organ that we do except for stomach and lungs. And they begin independently feeding around 5 days or 5 or 6 days post-fertilization. So because they're small and develop externally and rapidly, they've been used for toxicology screening studies in a variety of settings, and everyone's probably familiar with that. So another reason that we're using this model for this particular research is that zebrafish also contain complex resident microbiota that dynamically change over developmental time. And importantly, you can control the colonization status pretty easily of this model organism. So you can rear zebrafish to be azenic or microbe-free through early development, and you can conventionalize or add microbes back to azenic or microbe-free zebrafish just by introducing these microbes to the water. So you can add back diverse mixes of microbes, you can add back single strains to test different hypotheses. And this is illustrated in the figure on the right that is from our first paper in this area that came out last fall. So what we did is we derived zebrafish embryos to be microbe-free. And you can see in the top right, that's a zebrafish at 2 DPF or days post-fertilization, and it's sitting within the acellular chorion. And it basically develops in there until about 3 days post-fertilization when it hatches out, and incidentally, that's when the innate immune system comes on board. And what you can see is we derive these zebrafish to be microbe-free, and then we introduced back a single strain of fluorescently labeled Aeromonas varroni. And you can see that until the zebrafish hatches out, the microbes are unable to get to the organism, so they basically glom on to the outside of the chorion. Once the fish hatches out at 3 days post-fertilization, these microbes quickly colonize the pharynx, and within a day they have colonized the full extent of the intestinal tract, and that is, for this particular microbial species, its preferred biological niche. So colonization happens incredibly rapidly, and this is a system in which you can control the colonization status in a number of really interesting ways. So this is the experimental system that we've developed to test whether microbiota modifies the toxicokinetics or toxicodynamics of chemical exposures. And I'm not going to get into the nitty-gritty of the methodology, but I will point out that we start with 2 groups of zebrafish on day 0, the conventionally colonized, which are colonized throughout the entire experiment without any intervention, and then we derive a subset of those conventionally colonized animals to be microbe-free or azenic on day 0. On day 1, we come back and we take those azenic animals and we maintain a subset of them, and then the other half we recolonize or conventionalize with microbes that we harvest from our aquaculture facility. And then we will, if there is a chemical exposure, we'll commence exposures statically on day 1, carry that out till day 6, at which point they need daily media changes and feeding, and then we replenish chemical exposures daily from day 6 to day 9, and we run a number of endpoint analyses on day 10. So really the take-home is that at the end of this we have 2 colonized cohorts of zebrafish, the conventionally colonized always shown in red, and the azenic animals colonized on day 1 always shown in yellow, and these serve, the AC1 group serves as a control for the microbe-free azenic fish. So as I mentioned, when we started, we were quite interested in looking at the microbiota gut-brain axis, so we use behavior as a functional readout of brain development. This is a 48-well plate with 10-day-old zebrafish, 1 in each well, and you can see them moving around the wells, and we apply different light programs to measure their responsiveness to light changes. And I'm just going to show one figure from that Phelps paper that illustrates a couple key points. So in figure A, this is the distance moved every 2-minute period on the y-axis and the time in minutes on the x-axis. So the testing period consists of a 20-minute period, 10 minutes in the light followed by 10 minutes in the dark. And this is preceded by a 20-minute dark acclimation period. So zebrafish, very characteristically in the larval period, if they're swimming around in the dark and then the lights turn on, they will freeze. And you can see that here at the beginning of the test. And then they will slowly increase their activity over time. If the lights go off following a light period, they will startle and then slowly decrease their activity over time. So this is a very characteristic and very conserved locomotor response to light changes. So what we noticed is when we were doing kind of baseline characterizations of our colonization system is that there were differences in locomotor activity in the dark period when you compared colonized to microbe-free zebrafish. So the first thing to note in figure B, which shows the mean distance moved in the 10-minute dark period, is that there's no difference in the behavioral profile between the conventionally colonized and the azenic animals colonized on day 1, and this is the basically control-like phenotype in the red and yellow bars. And interestingly, what we noticed is that azenic animals, and again, these are zebrafish that lack microbes, were hyperactive relative to their colonized counterparts. So there was a phenotypic difference. And we were able to mimic hyperactivity that we observed in azenic animals simply by treating conventionally colonized zebrafish with broad-spectrum antibiotics. And so this was kind of important because this showed that you could apply a pharmacological compound or suite of compounds that are designed to kill microbes, and you could produce a phenotypic outcome. And so this really supported that toxicodynamic hypothesis that I introduced at the beginning. And it turns out in the case of the environmental chemicals that we've looked at, this doesn't really seem to occur quite very often. So I'm going to switch gears and head into our first chemical case study looking at triclosan. And we picked this compound. It's a antimicrobial compound. It's been phased out of some products in the past year and a half or so. And basically we use that same 3-cohort system where you have 2 colonized cohorts and your microbe-free zebrafish, and they're dosed on days 1, 6, 7, 8, and 9. And on days 6 and 10, we looked at 3 endpoints. We looked at the composition and the abundance of the microbes that are associated with the host using 16S rRNA sequencing. We looked at chemical metabolism using targeted and non-targeted analytical chemistry. And we looked at that behavioral endpoint I introduced. So this is a non-metric multidimensional scaling plot. This is a way to globally look at the overall community structure of host-associated microbes, and these are 6-day data, 6 DPS. So what I'm showing is the 2 colonized groups. In yellow is the azenic animals colonized on day 1, and in red are the conventionally colonized animals. And so basically what we're observing here is that there's a big separation between the 2 colonization statuses across this axis 1. And there, in the case of the conventionally colonized animals, there's really not much difference between the treated and controls in terms of the types of bacteria that are present and their abundances. And you start to see separation in the AC1 group, although again it's not consistent with what we saw with the other colonized group. And this is reflected in this heat map where we used an unsupervised clustering approach to ask how similar these community profiles are, community structure profiles are to one another. And this is showing family level taxonomy. And basically the thing to focus on from this particular figure is that the major discriminating factor in these data is not whether or not the animals were dosed with triclosan, it's what day they were colonized on. So on the left-hand side of the heat map are all of the AC1 samples, and on the right are all of the CC samples. So at 6 days post-fertilization, which if you remember back to the beginning of the talk is just 2 days after microbes get into the gut, the major discriminator in terms of who's there and how much of them are there is what day they were colonized, so what that initial inoculant looked like from the fish room. This really changes by 10 days post-fertilization when host-associated microbiota structure has begun to solidify. So here what you can see as well, along axis 2, there is still some separation between the DMSO vehicle controls for the CCs in pink and the AC1s in yellow. There is this convergence of global community structure in both treated colonization cohorts at both concentrations tested. So you really see a selection for triclosan-resistant, the same types of triclosan-resistant bacteria at 10 days post-fertilization. And this is reflected in the heat map. So as opposed to the 6-day data where status was the main driver, here it's whether the triclosan exposure is the main driver. So you have all of the DMSO controls on the left-hand side of the heat map, clustering, and all of the treated samples on the right-hand side. And there still is separation that is colonization dependent, but you can see these are more similar profiles than we saw at 6 days post-fertilization looking at family level taxa. So there's 3 groups roughly of taxa that we identified, and the first that I'll point out is this bottom bunch. So these are microbes that you might predict to see. So these are triclosan sensitive microbes that are present in our DMSO controls and that are inhibited or nearly disappear in their presence in the presence of the compound. And at the top of the heat map, there's a second class of taxa. These are microbes that are present to some degree in the controls, and they are selected for. So these are the so-called triclosan-resistant microbes. And the third class of microbes is shown in the middle, and this is basically an artifact of how we do the study. So they're present on day 0. They're present in only the CCs and not the AC1. So these are microbes that were unique to that day 0 inoculant. So it's an artifact of the experimental design. Okay, so we are able to select for triclosan-resistant bacteria in a reproducible way across both of our colonization cohorts. So we next wanted to ask whether those triclosan-resistant microbes are able to affect chemical toxicokinetics, whether that's uptake or metabolism of our xenobiotic compound. And at 6 days post-fertilization, the answer is no. There is, as you might predict, a significant effective dose, meaning that as the concentration goes up in the well, in the nominal water concentrations, you see a concomitant increase in the tissue level dose in the fish. And when we look at 10 days post-fertilization, where again we saw that really robust and reproducible change in host-associated microbiota community structure, we saw this really interesting chemistry finding. And this was kind of the opposite of what we were hypothesizing. So initially we thought we were going to see more parent compound in the azonic animals because the colonized animals would be able to biotransforming it into something else. And we obviously observed the opposite phenomenon, where here we're seeing a significant effective dose, a significant effective status, colonization status, and an interaction between both of these factors. So this is showing that animals that are colonized with triclosan-resistant microbes have higher concentrations of triclosan. So of course these microbes might be functioning as a sink and might be, chock full of the compound, and that might be explaining some of this elevation, but we also think that there is something to do that these microbes are also in some ways increasing levels of the parent compound in the host, and I'll get to that. So in collaboration with a bunch of neuroscientists, and I'm going to get into acknowledgments at the end of the talk, we wanted to ask whether these triclosan-resistant microbes could be biotransforming triclosan into unique chemical metabolites. And so the main biotransformation pathways are shown in Figure A that are related to both host metabolism and to microbial mediated metabolism. This includes a sulfation, glucuronidation, and methylation reaction. So the same samples that we used to perform targeted chemistry were also used in a non-targeted assay, and the suite of chemical features that were identified were screened against known triclosan metabolites, the DSS Tox database, and Metlin. And there were a bunch of features that came out of that went through a pretty strict filtering protocol to remove artifacts, duplicates, non-reproducible features, and that led us to a feature set of 563, which were further filtered. And the most important filter we applied is that we were interested in features that were at least twofold different. If you looked within status, so within the Azenix, for example, and you compared the DMSO vehicle control to the highest concentration of triclosan, or if you just looked at the highest concentration of triclosan and you compared the feature sets across statuses, comparing, for example, Azenix to the conventionally colonized. And when we did that, we then drilled down to try to find the parent compound. We were unable to identify it in the 6-day data set, so we focused instead on the 10-day data set, which again is where we saw that very conserved compositional change and the change in parent compound. So there were 78 features that met these filtering criteria at 10 days post-fertilization, and they're reflected in these volcano plots here. And so basically, the features that were significantly elevated in the conventionally colonized versus the microbe-free, or the azenic animals colonized on day 1 versus the azenic microbe-free group, are shown in the top right quadrant. And there's many more red dots than there are blue dots, indicating that animals that have microbes are generating more chemical features, as you might predict. And you'll notice the purple and lavender dots here, which relate to the parent compound in triclosan sulfate. And that data is shown on this slide. So what's neat here is that this is a totally different approach, and we were able to recapitulate the targeted chemistry findings, where we saw more triclosan in colonized animals relative to the microbe-free azenic animals. And when we looked at, just to give you an example of one chemical metabolite that we're focusing on, this is the sulfation reaction, triclosan sulfate. It follows the same pattern, where there seems to be more of this chemical metabolite in animals that contain microbes, and this suggests that it is the microbes that are performing this particular biotransformation. So this is a case study example supporting that toxicokinetic mechanism, where we've shown that the host-associated microbes seem to be capable of biotransforming the parent compound into chemical metabolites that have unknown toxicity profiles, like in the case of this sulfated triclosan. So the second case study I want to present gets all the way to mechanism. And this, when we first started this work, relative to performing these types of studies in mammalian systems where you have to maintain your breeding population sterilely, which is very difficult to do, and it's much harder, it's not impossible, but it's much harder to get all the way down to phenotype, I naively thought that it would be much more simple to get to phenotype using a zebrafish model, and this is really the first example where we have. So it's still not quite as simple as I initially thought it would be. So we're using the same exposure paradigm where we dose on days 1, 6, 7, 8, and 9, and on day 10 in the colonized animals we looked at composition, and in our 3-cohort system we looked at behavior and internal dose of the parent compound using targeted chemistry. And this was performed and led by Dr. Tara Katrin, a really excellent postdoc in the lab. So the first thing I'll show is that exogenous estradiol exposure did not affect microbial community structure. There's really no pattern to this NMDS plot on the left or the stacked bar graph on the right. And this might be expected when you consider that this is an endogenous hormone, so it kind of is a bit reassuring that the host-associated community profile is not really reactive to this particular compound. However, when you look at behavior in just colonized animals, we observed that increasing concentrations of exogenous estradiol produced hypoactivity in both the light and the dark period. So this is the phenotype that we started to get interested in. So Tara looked at behavior in the 3-cohort system, and this is a really busy figure, so I'm going to walk you through it. So on the top is that behavior data in the 20-minute testing period, shown every 2-minute period. And what you can notice is that in the case of the colonized cohorts and the azenic cohort, treatment with estradiol caused hypoactivity in all of the dark periods. So colonization status did not seem to do anything here. But when you look at the light period, it was pretty interesting. So, and this has been replicated. So this is a reproducible finding in a completely different study. You see hypoactivity, as I showed you on the previous slide before, that in the conventionally colonized animals, hypoactivity in the azenic animals that are colonized on day 1, and no effect in the microbe-free zebrafish. And we know increasingly from a number of chemical toxicology screening studies that this light-dark phenotype is incredibly complicated from a molecular signaling pathway perspective, and there's a lot of pathways that potentially feed into it, and it is quite common to see chemicals that produce effects that are specific to light phases. So what these data are showing is that to generate hypoactivity in the light period, microbes need to be present within the system. So we took a look at concentrations of estradiol in our 3-cohort system, and we saw something that was also pretty interesting. Now, this was the opposite of what we had observed in that previous triclosan case study. And here, what we're seeing is 3 times more estradiol present in the tissue of animals that lack microbes. And when you put it in context with the data that I showed you on the previous slide, our working hypothesis is that colonized animals are bioactivating exogenous estradiol into some sort of active metabolite that can produce hypoactivity. And we've performed non-targeted analysis. I don't have that data to share with you today, but we're trying to generate some ideas about what metabolites might be responsible for that colonization-dependent phenotype that we observed. So this is a case study or an example where we were able to show that microbes are affecting the kinetics of the exposure, and that leads all the way to a specific phenotypic outcome that relies on the presence of microbes. And the last case study that I want to share looks at bisphenol A and a number of BPA replacement compounds shown on the right-hand side of the slide. So this is bisphenol AF, BPB, BPF, and BPS, and this study was also led by Tara. So here, because we were looking at so many chemicals side by side and so many concentrations, it's a 6-point concentration response curve, we were only able to do this in a single colonization cohort, so all of this data is performed in conventionally colonized animals. We maintained the same exposure paradigm where they were statically exposed from day 1 to 6, and then daily exposure renewals on days 6 through 9. And on day 10, we looked at composition and behavior. So I didn't show the dose response or concentration response data for the first 2 case studies, but this is when we start off with new compounds, this is the first experiment that we do, which is basically to understand which, if any, concentrations of the test compounds produce malformations or lethality. And then we perform all of our subsequent studies using concentrations that do not produce malformations, because of course if you have a malformed animal and you see a swimming effect, it really doesn't mean anything. So I'm including the concentration response data here to illustrate a point that becomes relevant later on, which is there's a pretty wide difference in host toxicity looking at BPA in these replacement chemicals, where BPS is the least potent compound, so it did absolutely nothing to the developing larvae, even at the highest concentration tested, and BPAF in figure E was the most potent chemical. So we're able to rank order these test chemicals based on their ability to cause either malformations or lethality in the host organism. And then we connected this to our compositional analysis to take a look at the ability of these compounds to produce microbiota disruption. And because we were able to test so many concentrations of the chemical, we were able to apply a really unique microbial ecology statistic to this data set, and this was, I think, the first that I've seen example of applying this type of statistic to tox data. And basically what we did here is we had 4 replicates per group, and just the mean symbol is shown per group just to make this graph easy to get into. But basically for each DMSO control for each specific experiment, that's shown in yellow. And as you increase the concentration of the test chemical, like in the case of BPS here, you can draw a line from the DMSO control to the highest concentration. And by performing an A-NOSEM ordered R test, or to derive this ordered R statistic, this outputs a value between 0 to 1. And the closer to 1 this value is, the more concentration-dependent disruption of host-associated microbiota you are seeing. So if you look at the examples here, BPA in red, BPF in green, and BPS in brown, these are the 3 compounds that we identified that massively disrupt host-associated microbiota community structure. And to give you an example of compounds that do not, so E2, which we already saw, is in purple, BPB is in blue, and BPAF is in turquoise. And you can see that there's really no linearity in terms of the separation between the DMSO controls and the highest concentration. So we were able to globally identify 3 compounds that disrupt microbiota and 2 that do not. And this is probably a more classic way of looking at these microbiota community structure profiles using stack bar plots. So on the bottom in figures D and E, there's the examples of BP replacement compounds that do not perturb microbiota, and you can kind of see there's really no patterns there when you move from the DMSO controls on the left of each figure to the highest concentration of each particular test compound. But if you look at the top row, particularly if you focus on figures B and C, you can see pretty clearly that there is a decrease, a seemingly conserved decrease in the Burkholz-Dariaceae family taxon, and there's an increase in Chromatiaceae, and there's a number of other changes that are interesting to us. And if you compare that to the data shown in Figure A, this is another example of a compound that's disrupting microbiota, but it seems to be doing something that is unique to BPS relative to BPA and BPF. So you can put all these data side by side to try to identify commonalities using the unsupervised clustering and the heat map visualization that's shown on this slide. And what's interesting here is that all of the DMSO controls are more similar to one another than to any of the treated. And this is really important. This is just showing the highest concentration of each chemical. This is important because these DMSO controls, each chemical study was performed on a completely different day. So there were different microbes floating around the fish facility and colonizing these conventionally colonized animals. So there's that innate variability in the system that is reflected here, being overall somewhat similar across studies. And as I kind of alluded to qualitatively on the previous slide, BPF in the green diamonds and BPA in the red triangles cluster together, so they have a similar response. The microbiota community structure similarly responds to these chemicals. And BPS has its kind of own response, which is quite different from what happens in the presence of BPA and BPF. And so you can take these sequences or these putative bacterial identities that you get to from the sequencing approach, and you can use computational approaches to predict putative functions of these microbes that are selected for by chemical exposures, and then you can ask what's different about those functional or putative functional classifications. And this is a whole huge data set that I don't have time to get into. I just want to give you one example. So I showed you on the past couple slides that BPA and BPF seem to be doing the same type of thing to the community structure. They're selecting for the same types of resistant microbes. And when you look at the putative functional abilities of the microbes they're selecting for, they all contain this pathway, or they're enriched for this pathway of bisphenol degradation. So basically what's happening is that these microbes that are resistant to BPA or BPF exposure are able to harvest carbons off of the structure for biological processes relating to benzoate degradation. and chlorocyclohexane and chlorobenzene degradation. So that's, I think, really cool. And I won't show any more of the functional predictions, but I will just say in the case of BPS, it seems to select for microbes that have massive effluxing pathways, so they're able to pump this compound back out. So one of the interesting take-homes from the BP study is that if you rank order these chemicals based on their ability to disrupt or to cause malformations or lethality in the host, this is the rank order that you get where BPAF is the most potent chemical and BPS is the least potent. And it's basically an inverse relationship to the ability of these chemicals to perturb host-associated microbiota. So the idea here is that if the host organism can withstand higher and higher concentrations of the compounds, you're getting into the range where you start seeing microbiota community structure effects, where you start killing microbes. And from a hazard identification perspective and a prioritization perspective, which is how zebrafish in the developmental toxicology space is increasingly used, if you're looking at developmental toxicity of the host as your endpoint, you may be missing effects to the microbes. But what those effects are really not clear, and I'll come back to that point in a second. So just to summarize what I've shown you so far, we've developed an experimental system to test whether microbiota affect the toxicokinetics and or the toxicodynamics of xenobiotic exposures. And we use this system to characterize it to find this pretty interesting outcome, which is that animals that lack microbes are hyperactive, and that you can mimic this hyperactivity in colonized animals that are treated with pharmacological agents that are designed to kill microbes, and that's antibiotics. But in the case of environmental chemicals, you're never really able to fully wipe out all of the microbes that are there, so we never really saw that hyperactivity phenotype again. Instead, what happens in the case of triclosan is that the chemical exposure selects for resistant bacteria that we think function to increase the host parent tissue dose and perform a number of biotransformations, including that sulfation reaction. And in the case of exogenous E2, this is a really cool case study where we were able to show that the exposure to this compound does not perturb the community structure of host-associated microbiota, but it does result in a potential bioactivation event leading to a light-specific hypoactivity event. And finally, in the case of the BP replacement chemical story, we identified for the first time an inverse relationship between host toxicity and microbiota disruption, in part because we were able to test so many chemicals side by side at so many concentrations, and that's one of the real advantages of this system. So to put everything back in that framework that I introduced at the beginning of the talk, I've shown you two examples that fall under the toxicokinetic hypothesis, where these microbes are biotransforming exogenous agents, in the case of triclosan and exogenous estradiol. And I've shown you one example where application of a exogenous agent, antibiotics, causes a toxicodynamic effect, leading to hyperactivity in the fish. And I've included the BP compounds under the toxicodynamic heading, although we don't really have any evidence to support that right now. And that really leads me to the two outstanding questions that come out of this work, which we've spent the past 2+ years on. And the first that we really need to get a handle on is, do chemical-induced compositional changes affect other aspects of development? So in the case of that BP case study, we looked at one locomotor behavioral test. There are so many others, and there are other endpoints that are relevant to D&T within this model, and there are other organs and systems that are known to be influenced by colonization status, like the immune system. So, I don't think we can quite say what the effects of those compositional changes are yet, because we haven't fully scanned the organism to look at other endpoints. And importantly, might these changes that we're identifying in community structure predispose the fish to future insults? And this is something that was actually unified when you look at our triclosan data set, the BP compounds that did disrupt microbiota, and triclocarban, which I didn't get into. All of these compounds do one thing that is similar. They decrease the diversity of microbes that are associated with the host. And the reason that we have, evolutionarily speaking, that we have all these microbes in such a highly dynamic and complex system is that you need that functional redundancy. So, from human studies, we know that westernized cultures have reduced diversity, and that's been hypothesized to lead to a number of disease outcomes or be associated with them. So might chemical exposures during early development function to stabilize the system in a less diverse way that would then predispose the organism to a future insult, whether that's another xenobiotic or some sort of environmental stress? And that's really, I think, where we need to go next with this. And lastly, we really need to understand if the microbes are, as we've shown, performing chemical biotransformations, does this broadly affect chemical toxicity? And again, this means getting to phenotype and understanding what the correct phenotypes are to look at. So with that, I'd like to thank the members of my lab. Drake Phelps, who's now a graduate student at NC State, collected all the triclosan data that I showed, and Tara Catron, as I mentioned, ran the BP and E2 studies. I'd also like to thank our funding, including the Pathway Innovation Project Award, and there are a number of collaborators at the US EPA that have made this research possible. I want to thank Nicole Brinkman and Scott Keeley in particular. They are experts in the microbiome of water out in Cincinnati, and they have really helped us bring this sequencing and analysis pipeline into our data, into our research program. I also want to thank James McCord, John Sobis, and Mark Streiner for their help with developing the analysis approach for the targeted and non-targeted data sets. And I want to give a big thanks to Adam Swink, who is a research scientist in NHEERL's core, who performed all of the analytical chemistry for us, and Leah Wiemus, who is a postdoc in NHEERL, who helped with some of the E2 exposures. And lastly, I want to thank the folks in the Zebrafish facility for getting us embryos to do our experiments. So I'm happy to take any questions. Thanks.
Thank you so much. All right, I'll let Tamara have a sip of water here. And let's see, I think I saw... So to answer the logistical question that we got on the webinar, we do record these, and we will be posting the recording on the Communities of Practice website. So be on the lookout for that. If anyone, I believe I muted the line here, so I'm going to unmute that. If anyone in the Adobe Connect room wants to post a question in the chat box or speak up on the line, or anyone in the room, feel free to ask any questions you might have.
The leader has muted your line. To unmute your line, press #6.
Bacterium. So that is a bacterial species that we don't see very often in the zebrafish in the community that we have down in the fish facility. But it gets to this, I think, really interesting larger point that I didn't have time to get into, is that this system changes quite a lot day to day, week to week, month to month. So we've been tracking that. And we do see pathogenic bacteria that show up in our system. And I didn't get into this either, but it does seem like some of those low-lying, like pseudoloma-type pathogenic bacteria do often get selected for following chemical exposure. So another offshoot of this project has been to try to kind of control that variability on the front end of the experiment. So we've tried to isolate individual strains of bacteria from our conventionally colonized animals and generate a defined microbiota. So we're controlling what's going in on the front end to see if we can have more stable phenotypic outcomes. And that's a project that's ongoing that I'd be happy to talk to you about offline. But we are finding that it's really interesting to get control like neurobehavioral development. You need to colonize azenic animals with just a single strain of non-pathogenic bacteria by 6 days post-fertilization. So there's some sort of developmental requirement for colonization, and if you colonize them with a pathogenic strain of bacteria, you have abnormal behavioral effects that occur.
When it comes to bacteria colony, how the diversity of bacteria colony do you have to, how similar they to wild type?
Oh, yeah. Yeah, I'll repeat the question.
Yeah, thank you.
Susie, right? Yeah. Oh, sorry. Okay. So the question is, how reproducible is the microbiota in our system to what's present in the wild? Extremely different, as you might expect. It's a totally different set of population. So it's really cool. The fish are colonized, and their microbiotic community structure is dictated by what is in the water of our aquaculture facility, and that depends on, the temperature that you're keeping the room at, the water, how salty it is. So many environmental conditions affect what the potential inoculant could be, and it is unrelated to what is present in wild-caught fish. Yeah, so if you look at a OTU sequence basis, but if you're looking at the functional capacity of those microbes, nobody's really done that type of comparison.
Do you know anything about whether like composition or diversity matters more when it comes to?
Okay. Yeah, so the question is what matters more, composition or diversity? They're interrelated, so you can't have one without the other. And we're using computational approaches to approximate identity to predict putative functions. So this is an imperfect process, and really what needs to be done and what we're trying to do with some of these bacterial sequences that we've identified and we've maintained unique isolates is we're doing whole genome sequencing to try to really understand what the functional capacity of these microorganisms are. And in the case I can share of triclosan, we have identified a single strain of bacteria that compound selects for that we think is performing that sulfation reaction. So we've bought 2 strains of it, and we're going to test in a test tube and also in a monoassociated fish if we can produce the sulfated form of triclosan just by using a single strain of bacteria. Any other questions on the line?
All righty. I don't see anybody typing, so I think we're good. Thank you so much for presenting. I thought that was really great, and thank you everybody for your questions. And if you have any other follow-up questions, feel free to pass them along to me, and I will share them with Tamara. But thank you all so much, and hope you all have a good day.
The leader has disconnected. The conference will be terminated in 5 minutes.